Today, we highlight the SoCalGas STEMnet Student Research Fellowship Program awardees. Representing 5 campuses, these 6 students will share their work supported by the program. I would like to thank SoCalGas for their support of these talented students. Undergraduate research is a high-impact practice that has lasting effects on students' self-worth and future careers. This afternoon, each student has 10 minutes to speak on their research. We will entertain questions after the final speaker. I'd like to first thank Monica Aracon, Operations Analyst at STEMnet, for coordinating this event today. Our first speaker is Trent Ruiz from CSU Channel Islands, and his research title is Computational Aspects of Peridynamic Corrosion Damage Modeling and Scanning Vibrating Electrode Technique Validation. Trent.
Thank you. Thank you very much. Let me share my screen here. Wonderful. And let me share my slides. Excellent. Thank you for such a wonderful introduction. And so as the introduction suggests, I'm going to be discussing the computational aspects, what it takes to simulate corrosion damage, and the physical experiment that we're going to be using to validate our computer models. And so at this point, you might be asking, well, okay, corrosion, right? But we're talking about gas, right? And it actually has more application to natural gas and active oil pipelines than one might initially consider. In April 2022, the Journal of Gas and Science Engineering published an article reviewing failure mechanisms related to oil and gas pipelines and found, perhaps unsurprisingly, that steel is the most commonly used material. And let me tell you, it's pretty darn corrosive. And being in Southern California, especially where I am right now, I'm about a mile from the beach. Even our very humid air can cause corrosion on our cars. I've had plenty of garden tools I've left outside that have not been usable after experiencing our salty, humid conditions. Now, let me ask you this. What do you think happens when we run these pipelines in salt water? In the Gulf of Mexico alone, there's 8,600 miles of active oil and gas pipelines. And when a metallic object is placed into a solution with a salinity similar to ocean water, corrosion is significantly accelerated. And so to investigate corrosion, we consider electrochemical corrosion. And there's many types of metallic corrosion, but most are electrochemically driven. If we draw our attention to the figure I have on the screen right here, we notice the cathodic and the anodic regions. We have an electron path and the ionic path, which are propagated by an electrolyte. Again, this includes very humid air. Under these conditions, the anodic region begins oxidizing, leading to corrosion and metal loss. The electrons that are flowing through the anodic regions then travel to the cathode, and once lost to the cathodic region, oxygen in the surrounding electrolyte reduces and reacts with the oxidized metal to produce metal oxide, commonly known as rust. And these electrochemical models work so well because of the insight that is gained in the proportional relationship to current density and oxidation. And so now we get to talk math, and it's one part I really like. And to implement these models, we have to summon the power of peridynamics. And it's a recently new branch of classical continuum mechanics, early 2000s recent. So it's pretty new, which treats objects as a continuous mass rather than discrete particles. And this is actually pretty huge, as previous models seeking to model such corrosion maintain costly iterative meshing and remeshing schemes, which also happen to be discontinuous at the crack surface. Oh, and it also doesn't allow for natural crack propagation. And so we've developed a peridynamic model that pulls from the two main thought colleges, the bond-based and state-based approaches, and we use a Poisson ratio to navigate between the two. We define a peridynamic horizon H, and it is within this horizon distance delta that a peridynamic node may interact with any other peridynamic node. In this case, it is interacting with, say, X prime, which forms our peridynamic bond. This image shows peridynamic graphical representation of peridynamic deformation. We have C, which is the relative position between nodes connecting a peridynamic bond. And we have eta, which is the relative displacement in peridynamic models. Both y and y prime exert an equal and opposite force on one another. And when this peridynamic bonds extends beyond the horizon, it is considered broken and can no longer exert any force. Since peridynamics avoids the pesky partial differential equations that have discontinuities at the crack surface, the integral nature of peridynamics allows for natural crack propagation. And so to simulate degradation, we are using the Laplace equation, and this equation governs the electric potential, and a consequence of which is electrochemical damage, electrochemical degradation. This couples very well with the state-based model where theta is our stiffness degradation and the kernel kappa is our bond stiffness tensor. To model damage due to corrosion, we are utilizing Perifast. It's a 3D pseudo-spectral peridynamic MATLAB code for corrosion. It was developed quite recently by Florent Barbaroux as an homage to him, an alum of the University of Nebraska. The sample code shows pitting corrosion on the University of Nebraska N. What we are looking to change is how it treats current density. The simulation looping on the screen treats current density as a constant. And we actually know that that current density is variable within the solid, especially near the pitting point. And so since it is an open source contribution, we will also be contributing to the online GitHub community with these adaptations. To verify these computer models of corrosion, we're going to be implementing a SVET experiment, and this measures current densities above a corroding surface. Industry standard machines use costly piezo electrodes to basically vibrate the electrode. The apparatus that is on the screen here substitutes the piezo with a solenoid, and to keep the solution space wide enough, an electromagnetic tactile transducer will also be explored. The solenoided transducer techniques will be compared to industry standard current density maps. In this diagram, we see the vibrating probe, which is connected to the solenoid or the transducer, depending on which experiment we're running. We have cameras to aid in visual inspection. Current density maps look quite a bit different, especially in the early stages of corrosion, than the object that is perceived by our eyes. I took a broken down 3D printer, it was actually 4, parted them together and removed the extruder, which has now become our XYZ plotter for the probe. It utilizes an Arduino Mega stepper motor controller, which allows for direct communication with MATLAB. MATLAB will allow for the acquisition of data. And we will use software-based lock and amplifier principles and Fourier analysis, which has been proved successful in previous research by Dr. Rasnow and his team on E-fields of weakly electric fish. This effectively raises the noise floor and gives us current densities that can be measured. The computer also maintains analog to digital converters and external instrumentation amplifiers. This was the actualization this semester of the previous diagram. We have 2 cameras. A solenoid is attached to an electrode, and this is the platform that we can substitute the transistor or a transducer into. Here's the electrolytic cell with a piece of metal encapsulated in platinum pure silicone, allowing only for the surface of the metal to be exposed to the electrolytic solution. The SVET apparatus will scan over the surface of the corroding metal and measure the equipotential surfaces that are permeating from the corroding metal. To drive corrosion, we're utilizing a power supply built from a previous class and additional drive circuits allowing us to oscillate corrosion. Here is an image of industry standard current density maps, and we will be comparing the maps that we get from this experiment two ones like this. This is from the VersaScan SVET apparatus. The darker pink regions are anodic sites where pitting is most likely to occur, and it shows corrosion is non-diffusive, and that current density does change over and through the surface when subjected to electrochemical corrosion. To view these models, we're going to be utilizing a new emerging technology, augmented reality. We very graciously got a grant for some Microsoft HoloLens 2s, which will allow us to augment our space. It allows us to view these current density maps and the corrosion simulations in 3D, giving us greater insight into the effects of corrosion. In addition, we're going to be also providing two-dimensional maps that can be viewed on a conventional computer. The future work for this summer is quite exciting. We're going to be exploring the effects of microbiologically influenced corrosion. 3 microbes in particular that we're looking at is B. subtilis, P. florens, and P. lipolytica. The first 2 actually inhibit corrosion, and the last one promotes it. And so the way that this works is that in like aquatic situations such as the ocean, when corrosion begins, a biofilm forms, and this does have the potential to increase the deleterious effects of electrochemical corrosion, and it's something that we want to be able to implement into our models. With the SVET apparatus, by the end of the semester, actually, we should be able to calibrate the vibrating probe, and we do this by creating a parallel plate capacitor and measure the known equipotential surfaces within that capacitor. And we will also be, after generating those current density maps, we're also going to be implementing the transducer technique. And with respect to Parafast, we're going to verify the model with the physical experiment. And then we're also going to be exploring aerospace application. Earlier this semester, I wrote a grant for NASA's Undergraduate Research Challenge, and it was accepted a couple weeks ago. And while the pressure is on, we're going to be focusing on aerospace materials manufacturing, next-gen aerospace materials manufacturing, but it shows the breadth of our work and how it can apply both to infrastructure and also these next-generation solutions. And as I'm wrapping it up here, I do want to thank everyone here. I want to thank everyone that put in all of their effort to make this STEMsnet conference possible. All of my wonderful mentors, Dr. Cynthia Flores, Dr. Gregory Wood, Dr. Brian Rasnow, Dr. Carlos Nash at UCSB, everyone in the research teams that I get to pull from wonderful knowledge from. Thank you so much. It is you guys that are making undergraduate research possible. Thank you.
Thank you, Trent. Our next speaker is Naomi Garcia from Fresno State, the analysis of the environmental growth conditions affecting algal, biomass, and lipid accumulation. Naomi.
Yes, thank you for that introduction. I'll go ahead and share my screen. Okay, so yes, this will be about algal, biomass, lipid accumulation, more specifically 2 environmental growth conditions that affect this. So let's get straight into this. Okay, so why are we looking into algae as a potential biofuel? So this organism has certain notable qualities. It's very clean and renewable as a resource. It can grow on non-arable land, and compared to other carbon-based counterparts, it just grows very quickly and doesn't take up much space. When you put algae into wastewater, it also has the potential to absorb the dissolved nutrients suspended within it. So aside from this, algae can also very nicely store lipids, which are also known as fats and oils. And this is very crucial when we interpret it as a potential biofuel, because those are possible sources of energy that can be exploited. So aside from this, it's just in general very nutrient rich. It can even be used as an animal feed and a food additive. So the 2 species that we will be working with is Chlorella prosthidicoides. I will be referring to this as CP25. And then down here we have Chlorella vulgaris, also known as CB. So in order to actually have optimal growth conditions, we need to look at a few factors. First of all, we want to make sure that we are increasing nutrient starvation as much as possible, that we have the right light intensity, and we can also really look at salinity and aeration factors. But overall, we're just trying to maintain a very delicate balance between all of these variables. So in terms of the actual experimental objective, we are specializing in two of the conditions or two variables. So the first is light conditioning. So according to past research, the red and blue LED forms of lighting actually were shown to lead to higher concentrations of lipids along with a greater biomass overall. When we look into nutritional availability, there's a very interesting relationship happening. So in terms of nitrogen-based nutrients, decreasing their availability was found to accelerate the lipid accumulation rates and biomass. But for the carbon-based sugars, basically increasing their availability will in turn accelerate the rates of lipid accumulation. So this is essentially happening because the algae cells, they start to really stress out and start to panic when they realize that the media they are suspended in doesn't have enough nutrients. So they will thus store as much energy as possible to supplement for this. So in order to address these hypotheses, we will be exposing two strains, the CV and the CP25, to all of these variables and all these standards, specifically using standard Hawaii media, since it already addresses the nutrient availability factor by not having the nitrogen suspended within. So essentially what we did here when we conducted experiments, we grew either mother cultures of CV or CP25, let them get concentrated for a few days, and then we inoculated in 3 of these different bioreactors for a period of 7 days. So initially we looked into the UTEX bioreactors, and these bioreactors up here. We could play around with 4 different lighting conditions, the blue, red, green, and white lighting. But with these, we had a huge contamination risk because they had portholes that were open up to the environment. So down here, we have the algal bioreactors. And with these, we had 2 milliliters of the CV and 400 milliliters of the Hawaii. One of them had glucose, and then the one next to it had no glucose. And overall, we had low contamination with these because we were not even having to take samples every other day. So there was less opening the culture up to the environment to get contaminated. And then we also generated additional growth curves with the growth chamber apparatus, as we see in the lower right-hand corner. So we had 1 culture with glucose, 3 without glucose. We had the same ratio of CV species as to Hawaii media. And we had medium contamination risk because we did have to take more samples in the run. So here is the data for the UTEX bioreactor. The first trial, we can only do 1 of them with the glucose. So as we can see here, a lot of the data is very difficult to interpret because for this trial, we had huge rampant amounts of contamination. So even the absorbance values were a little bit disturbed because the cultures actually shifted to a light orange color, which we'll see later on. So the one thing that we could take away from this data, though, was that the green culture specifically had a much higher cell count in comparison to all the other cultures. And this was also reflected in the total biomass that we see displayed in the graph below. So green just has a lot more cells. So the bulk of the data for this research came from the algal bioreactor trials. We had the white, red, and blue lighting growth curves that we have right here. These are for absorbance. And the reason they look different from the other absorbance growth curves is that for the algal machines, they are able to take absorbance readings pretty much continuously throughout the 7-day period. So when interpreting these graphs, we see that the no glucose cultures for all of them seem to have much higher absorbance values. Now, when we look into the white and the blue lighting conditions specifically, we can see that this is juxtaposed by the fact that the glucose cultures had higher cell counts and biomasses despite having lower absorbance growth curves. So for red lighting, however, we see that the 2 growth curves are extremely close together and much higher in absorbance in comparison to the other ones. And for this specific lighting condition, even the no glucose culture retained a higher total biomass and higher cell count. So this is a very interesting trend to note. So here is the data for the growth chamber. We initially just ran the growth chamber experiments just to generate more growth curves and just to reiterate the nutrient starvation factor. So here, when we look at these graphs, we have the 3 glucose cultures, and they pretty much excel in everything. So they had higher absorbances. For cell counts, there wasn't much of a difference between that and the no glucose. We can still see that they had higher cell counts to the no glucose cultures. And this was also reflected in the total biomass. Very notable to point out, though, that the glucose 2 culture did have a significantly higher biomass as compared to the other glucose cultures, though we did use the same standards for all of the glucose cultures. So another very interesting data point. So then we have the Boda-P staining that we performed. This is also known as fluorescence microscopy. So essentially, we dyed all of our algae cells to really visualize those lipid masses that we expect to find in there. So in terms of the growth chamber graph here, we see that around days 5 and 6, we really start to see a pickup in the accumulation of both lipids and an increase in cell counts. The no glucose culture is a very interesting figure to look at because we see that there are a lot less cells in the images, but when we compare the algae cells to the other ones, they seem to glow brighter and they seem to be larger. And that's also apparent when you look at the scales at the bottom. They're 50 micrometer scales. So overall, these algae cells did seem to be accumulating more lipids. So that's very interesting. So next to that, we have the algem images. Though there wasn't much of a difference and we weren't able to get data for the red lighting conditions, we do see that for the blue light, we have a lot more cells there, and even the glucose blue light sample for day 7 seemed to have brighter glowing cells, so suggesting that they probably accumulated more lipids. All of this is juxtaposed by the CP25 Mutex bioreactor for the green lighting on day 7, in which we see so many more cells, which was incredibly unusual. And we can actually very clearly see those small little circles inside, or the lipid body masses in there. So when we are addressing the conclusions, in general, the samples with glucose and yeast multiplied greatly, but they didn't have the brightest cells or highest absorbance values. In terms of nutrient starvation, also, we couldn't really address that with the bioreactor trial 1 data, but we did find out that the green light, it thrived despite contamination. In terms of lighting intensity, the algem bioreactors, the CV inside did seem to have an affinity for the red lighting conditions, but in terms of growth in general, all of the CV in the growth chamber exposed to fluorescent lighting did support much denser cultures than any of the algem lighting conditions. So that's something to look forward to in the future. Speaking of future direction, what we have planned to do is, in general, we need more algem runs for CP25 instead, because the other ones were only with CV. We need a better exploration of the trophic conditions, and we actually need to perform the FAME method of analysis to see the actual concentrations numerically of lipids in each of the samples, as well as what types of lipids were present. We need to revisit the UTEX bioreactors to find possible sources of contamination, and in general, repeat as many trials as possible to find more definitive results. So some key research takeaways. Initially, when I found out that the first bioreactor samples were contaminated, it was very disappointing, and I thought that that data wouldn't be able to be used. But I did learn that there's no such thing as a failed experiment. So in order to change the trajectory of an experiment when things are not going right, I learned that adaptability is key. You need to collaborate with peers as much as possible, because chances are that they know something that will be able to help the trajectory of your experiments. So I would like to thank the STEMnet SoCalGas Research Fellowship Award for this wonderful opportunity. CSU Fresno, more specifically, the College of Science and Mathematics Chemistry Department, my mentor, Dr. Mitra, and my researching peers. This is my first semester doing research, so none of these experiments would have been possible without the generous assistance of my peers. Thank you.
Thank you, Naomi. Our next speaker is Ysab Espinosa from Cal State San Bernardino.
Thank you for the introduction. I'll share my slides. Okay, so this semester I did research on trying to find an annual meteor shower on Mars. And we use data from a spacecraft called MAVEN to try and look for unique phenomena such as meteor showers. And there's more than 8 years of data to look through. We're hypothesizing that meteor showers on Mars could be linked to high up in the atmosphere and that they impact Martian atmosphere dynamics. And this is important because it could potentially help spacecraft safety if we watch out for things in the atmosphere. And it's also relevant if we could find something about how meteor showers happen on Mars, it could be applied to other planets as well. So where do meteor showers even come from? They come from comets that come from the Oort cloud. When comets burn up in their orbit, they leave behind a dust tail, and that dust tail will possibly be within the orbit of a planet. And if that planet goes through the dust, it'll burn up in the atmosphere and create what we see as a meteor shower. If this happens, it's going to go through the same spot every year where the dust was left behind by the comet. So a meteor shower could potentially be annual, so it'll happen every year, either on Mars or like we see on Earth. Just this past weekend, there was the Lyrids meteor shower that you might have seen. On Earth, we can see the meteor showers coming down as what we call shooting stars. But in this research, what we're looking for isn't shooting stars. We're looking for concentrations of metals in the atmosphere that aren't normally found there that can hint that the comet dust was there. An example of this that we've found before is Comet Sighting Spring. It's a meteor shower that was detected on Mars by MAVEN using the IUVS technology, which looks for how many photons per cubic meter there is of metals found in meteor showers, such as Mg+ or iron. So far, Comet Sighting Spring is the only shower to be detected on another planet. And Comet Sighting Spring doesn't make an annual or a yearly shower like we're looking for, because it only has passed through the solar system once, but we're looking for something that does happen every year. You can see here on the pictures that we're making these graphs comparing scan to altitude. And the color in these pictures you can see is how much of specific elements are present. So brighter regions will signify that there's more of a certain element, and darker regions will show that there's less. So using this, we're going to look for examples of time periods where there's a lot of high concentrations of the metallic elements such as Mg+ or iron, and hopefully find something. So yeah, like I said, we're looking for something different from a quote unquote normal day on Mars. A normal day on Mars will look like the picture you see here, where there's not super high concentrations of magnesium. And it's not super high up, it's around 100 kilometers. During showers, you're gonna find that the higher concentrations of magnesium are gonna be found in higher altitudes, but it's unclear whether or not it's gonna be particularly denser. You might expect a faster meteor to sort of punch through the atmosphere and make it down farther, but it actually happens the opposite way, where a faster meteor will ablate higher up in the atmosphere because it reaches an orbital velocity. So far, we haven't found another meteor shower quite like Comet Sighting Spring, but there's strong evidence that we're headed in the right direction. So now, looking through the data that we've compiled into images that make it easier to look through, we created a rubric for how we can determine if this is an example of something where a meteor shower is happening or whether it's not happening. So we're going to call a day where there's almost no magnesium deficient. A normal day on Mars could be called fair. Good is when you're going to be either higher in concentration or altitude, either one or the other. And super, which is possibly a meteor shower, is going to be when you're higher in both concentration and height. So here's an example of something we're going to call deficient. There's another on the left, you can see scan versus altitude again, and everything's pretty dark red, which shows that there's not very high concentrations of magnesium. And to the right, there's this other picture we can look at that compares concentration of magnesium to altitude. And it also has extra information like latitude, longitude, and importantly, the Ysab, which tells you what time of year it is on Mars. Here on the dot plot, you can see everything's pretty much to the left around 0, which also shows low concentration. So this is a deficient example. An example of a normal day on Mars would be something more like this, like we saw before. Yet again, there's a little bit more oranginess around 100 kilometers, but nothing super high in concentration. And in the plots, you can see the concentration gets a bit higher, but nothing particularly significant or high up. Once we get into the more good category, you can see we're either going to be higher in concentration or altitude. Here it's higher in concentration. So the values of the concentration of the magnesium you can see on the plots get up to 500 or almost 500. And on the picture, there's some yellow areas and some bright white, which shows that there's a lot more magnesium, but it's not super high up. It's hanging around 100 kilometers up from the surface of Mars. Here's an example of good again, where you're higher in altitude this time instead of concentration. The numbers don't get quite as high as the last picture, but you can see that on the right, the peaks are around 140 kilometers this time instead of 100, which is higher up. And on the left, you can kind of see the yellow bits are up higher, but that they're not quite to the white in color as the last one was. So those were the examples of good. And lastly, the example of what we call super would be when we get these yellow and whites high up. So we're higher in concentration and altitude. So this could be a possible meteor shower that happened because these metals were present that aren't normally present. And you can see the Ysab telling us what time of the year it is. So if this is a possible annual meteor shower, it could happen yearly around this time of year on Mars. Here's another example of a super quote unquote picture where yet again we have whites and yellows around higher altitudes. And we can see that the Ysab here is similar, which is a good sign. Moving forward, we're trying to organize the data by its Ysab and categorize it so we can try and find trends that might lead us to a meteor shower. And we're trying to make a timeline so we can look through multiple years and see if a pattern happens where a possible meteor shower happens at the same time every year. Right now, we're getting tantalizing hints pointing towards Ysab being between 320 and 330. So hopefully, as we continue looking, this trend will continue and we might find an annual meteor shower on Mars. In summary, we haven't found another meteor shower like Shining Spring quite yet, but we can use the tools we made this semester to keep looking through multiple time periods and find some consistently high magnesium concentrations at consistently high altitudes. So in the future, we can see what happens. I want to thank Dr. Krismani and STEMnet for allowing me to do this research this semester, and thank you guys for your time.
Thank you, Isabel, for your presentation. Our next presenter is Ernesto Chavez from Cal State Long Beach. Plant traits that promote successful establishment when restoring degraded SoCal shrublands.
Hi, good morning. Thank you for that introduction. Good morning, everybody. My name is Ernesto Chavez. I'm from California State University, California State University, Bakersfield, and this is plant traits that promote successful establishment when restoring degraded California shrublands. Before I get started, I'd like to give some quick thank yous to STEM SoCalNet, to the CSUB RCU mini grant, to my mentor, Dr. Brandon Pratt, and my research associates, Dr. Anna Jacobson, Antonio Materio, Dr. Emma Underwood, Dr. Nicole Molinari, Shonda Weiss, and to the Santa Barbara Botanical Gardens. So chaparral is a vegetation type that is prevalent in California, and it is actually the most extensive vegetation type here in the state. It occurs in low to mid elevations, and it is prominent in the Mediterranean type climate. Mediterranean type climate is categorized by cool, wet winters and hot, dry summers. Chaparral is an evergreen shrub, meaning that its leaves are green year-round and they do not shed seasonally. Its leaves are also sclerophyllous, so they are thick and leathery. And chaparral is also prominent in fire-prone ecosystems. Coastal sage scrub, on the other hand, is a different vegetation type, but it also grows alongside chaparral, and it also occurs in lower elevations, and it is prominent in the Mediterranean type climate as well. It differs from chaparral, however, in that its leaves are dimorphic, meaning that it sheds them seasonally, and they are also soft and aromatic leaves. On the right here, you see a picture of coastal sage scrub, which during the wet season, which you can see the shrub is very green, but this changes during the dry season, and it sheds its leaves and becomes much more drier and loses its color. So these shrublands have been subject to degradation and transformation in the last several years. Landscapes have transformed from dense closed canopies like the one we see on the left to much more open landscapes that have been invaded by exotic annual herbs. Some of the causes for vegetation type conversion include drought, fires in rapid succession, introduced species and pathogens. And vegetation type conversion causes many changes in the ecosystem of these landscapes, and it changes the composition of plants, animals, and microbes in the ecosystem. And this is also a permanent state. So to restore these landscapes, active restoration is needed. And this has been the main objective of the restoration project taking place at Lake Piru in Southern California, just some distance north of Los Angeles. These are some of the restorations plots that we have been working with, and some of the species that we have also been working with. For this project, we focused on 9 species in total, 4 of which were chaparral and 5 were coastal sage scrub. The chaparral, we used Cyanotus eleganthus, Heteromeles arbifolia, Frangula californica rustivata, and for coastal sage scrub, we focused on Artemisia californica, Baccharis pilularis, Euryogonum, fasciculatum, Hazardia sclerosa, and Celia californica. While we were working with this restoration project, we noticed that some species were better established and seemed to be growing and having higher success rates in restoring than others. As you can see from this graph, we had some plants that had very low mortality rates and some that were on the complete opposite of that spectrum and had extremely high mortality rates to the point where they were about at 100%. And we wanted to figure out why this was happening. So this brings us to a pretty important point of acquisitive and conservative traits in plants. Acquisitive traits would be more associated with higher growth rates in plants and larger plants because they would typically use their resources as they get them. Conservative plants, on the other hand, would be associated with slower growth rates and smaller plants as they would conserve their resources for when resources are scarce. For this, for my study, I hypothesized that plants that have traits associated with differential resource use strategies would help predict mortality patterns. I also predicted that plants with that could switch between more acquisitive traits when resources are plentiful and switch to more conservative traits when when resources are scarce would be more successful in restoration. To predict these species mortality patterns, we took measurements from many functional traits from published and unpublished data, as well as some that we measured on our own. Some of the traits that we measured were specific leaf area, which is ratio of leaf area to leaf dry mass. We also measured maximum height. We measured growth rate, which is height change over time, leaf nitrogen concentration, fluorescence, and P75, which is loss of productivity at 75%. So the general trend that we saw with growth rate was that as growth rate increased, mortality decreased, and we saw a strong association with this. We saw that typically chaparral shrubs were had a lower growth rate and increased mortality, and coastal sage scrub were typically on the higher end with higher growth rates and less mortality rates. We also saw something similar with leaf nitrogen concentration, and chaparral were also much more prone, or much more were on the smaller side of leaf nitrogen concentration, and were had higher mortality rates compared to the coastal sage scrub. We also saw this with fluorescence. Coastal sage scrub tended to do better in this case too, but we did see a bit more of variation between some species. We also saw this again with specific leaf area, and we saw that as specific leaf area increased, mortality also decreased. Again, the chaparral were on the higher mortality rate, and they had a smaller specific leaf area. For P75, again, we saw something similar with as the values were less negative, the mortality rate tended to decrease a bit more. Mortality rate was decreasing as well. And we did see a bit more variation with this, as we see the chaparral were more scattered throughout the plot, but coastal sage scrub was more consistent with what we were seeing previously. For maximum height, we did see an inverse relationship, and we see that as maximum height increased, mortality also increased. Again, chaparral were on the higher end of that of the height spectrum, and they had higher mortality rates as well. So here we put all of the traits we measured in one graph, and we see that in a principal component analysis graph, and we see that leaf nitrogen mass, specific leaf area, growth rate, and fluorescence were strongly associated with one another. And inversely to that, max height was inversely proportional to that. We put that again on a linear model, and we see that there was a strong association between these traits and mortality rate. We also see between species that growth rate, that coastal sage scrub typically did at a significantly higher growth rate compared to chaparral. Also, we see that coastal sage scrub also had a significantly higher growth rate, or sorry, significantly higher specific leaf area than chaparral. Same when it comes to maximum height, although we did see an outlier with coastal sage scrub. And overall mortality wise, we did see that coastal sage scrub typically did way better than chaparral. So in this study, we found that plants with higher growth rates, leaf nitrogen fluorescence, and specific leaf area were strongly associated with lower mortality rates. Coastal sage scrub had many favorable traits and had a higher success rate in comparison to chaparral. And for chaparral to be successful in restorations, it would likely need further aid and some kind of facilitation to increase the survival rate. Thank you.
Thank you, Ernesto. Our next speaker is Ethan Luksik from Cal State Long Beach. Title of his talk is Biodegradable Metal Organic Framework Thin Films as Drug-Eluting Stent Coatings.
Awesome. Thank you for that introduction. Let me just share this screen real quick. Okay. Cool. All right, so I'm a part of Dr. Feng Yuantian's research lab here at Cal State University Long Beach. And our lab focuses on working with metal organic frameworks and their applications to cardiovascular diseases. So today I hope to share a little bit about what goes on in our lab and some of the exciting results we've gotten in the past few months. I wanted to start by talking about what metal organic frameworks are, since that's the majority of the research I will be presenting today. Metal organic frameworks, also called MOFs, are crystalline materials constructed from metal ions bridged by organic ligands. Metal ions will bind to the nucleophilic regions of these ligands and form porous structures that can fold three-dimensionally. And these MOF structures stack on top of one another to form crystals of different shapes and sizes, as shown in the top here. And these are some electron microscope images of the types of crystals that we actually grow in the lab. So the overall goal of this project deals with arterial heart disease, and more specifically atherosclerosis, which describes the buildup of plaque in arteries, and focuses on how we can use metal organic frameworks to combat this buildup. So plaque forms when cholesterol lodges in the walls of our arteries, and a modern fix for this plaque buildup is known as balloon angioplasty surgery. And many of you may know this procedure or have family or friends that have had this done. It works by inserting a drug-eluting stent, abbreviated DES, on a collapsed balloon into an artery, inflating the balloon and expanding the stent, and then deflating the balloon and removing it to leave the stent structure to support the affected conduit. The stent is a polymer-coated steel structure that is collapsed when inserted and can expand with the balloon. But the problem with these commercially available drug-eluting stents is that they have a disadvantage in working long-term, as their polymer coatings are linked to late stent thrombosis, and that's shown in the bottom image here. So late stent thrombosis is where the body rejects the stent due to poor biocompatibility, which causes hypersensitivity, impaired healing, and plaque reformation. So our project hopes to replace that polymer coating on the drug-eluting stents with MOFs loaded with ibuprofen to deliver anti-inflammatory drugs to arterial passages that have recently undergone angioplasty surgery to heavily reduce or even prevent the occurrence of late stent thrombosis. So my specific contribution to this project revolves around the iron and terephthalate-based metal organic framework called MIL88B. So MIL88B in specific is an ideal MOF candidate for potential stent coatings due to high biocompatibility. The degraded products of MIL88B include terephthalic acid and iron ions, and they exhibit no adverse effects in humans because you all have iron and terephthalate in your blood. So in addition, MIL88B has excellent drug absorption capabilities with regards to the size of anti-inflammatory drugs such as ibuprofen. Recently, our lab has been working on stability tests on MIL88B, and these stability tests will help us determine the rate of degradation of this MOF and its sensitivity to different environments. So the stability tests of MIL88B measure the degradation rate of the framework in different pH environments. And the reason we're doing tests on pH is because the pH gradients in the body have one of the most significant contributions as environmental stimuli for the triggering of drug release. So for instance, the gastrointestinal GI tract contains a broad range, a pH range between 1 and 7.5. And this can result in various retention times of therapeutic compounds within the body. So to test if MIL88B is pH sensitive, QCM was used to test and monitor its degradation rate in different pH solutions. The QCM apparatus is shown in the middle and depicts a gold chip coated with MIL88B crystals, which have been loaded with ibuprofen and are left in a buffer solution of the predetermined pH that we are testing. The apparatus is left running for a duration of 48 hours as the MIL88B crystals begin to destabilize and degradation occurs, releasing our anti-inflammatory drug into solution. The gold surface in the QCM setup acts as an inert conductor for the QCM to actually read the degradation rate of MIL88B, and the gold surface doesn't react with the MOF, but allows a current to be run through the sample. So a QCM is a quartz crystal microbalance, and this is designed based on the piezoelectric effect of quartz. So when a current is applied to a quartz crystal, it oscillates at a known frequency, and the frequency can be slowed with a mass on top of the quartz crystal. So the data from QCM can be graphically portrayed using the Sabare equation, where delta M is the change in mass per unit area, delta F is the observed frequency change in hertz, and CF is the sensitivity factor constant for the crystal. So as the frequency increases, mass is lost, meaning that MIL88B degrades and releases ibuprofen into solution. From this, we're able to determine the degradation rate in different pH environments. The graph at right depicts a real-time QCM measurement of ibuprofen released by MIL88B at a pH of 6.3, and that's the red one, and pH of 7.4, that's the black one. So at a pH of 6.3 in red, we can see that the frequency increase tells us that there is more mass lost at this lower pH. So the data from QCM analysis after applying the Sabare equation tells us the method of degradation of MIL88B in the chemical environment in which we are testing. So there are three methods by which MIL88B degrades in solution. The first level of degradation is diffusion control and is a zero order reaction. Here, ibuprofen diffuses out of the metal organic framework structure into solution, leaving rapidly. The second level of degradation is surface erosion. And in this decomposition, the surface of the MOF is exposed to solution and is degraded first, causing ibuprofen, iron, and terephthalate to degrade into the surroundings. And the third level of degradation is bulk erosion. And this occurs by first order. Hydrolysis causes larger structural erosions of MIL88B, releasing both ibuprofen and MIL88B into solution. So from the QCM data and characterization using X-ray diffraction, we were able to determine that MIL88B is pH sensitive. And this data can support future research in determining the optimal pH for MOF drug release. And more importantly, it confirms the potential use of MIL88B as a carrier for ibuprofen on drug-eluting stents. This data demonstrates the viability of applying these MOFs in a real-world medical environment. And there's a paper that was recently published by our lab following the conclusion of the stability test. So we were excited to get that research out there. So I wanted to show this slide really quick because I wasn't going to have a lot of time to get into this, but these are some of the experiments that we have been designing for the future of MIL88B. And this series of experiments hopes to explore a faster synthesis method utilizing electrophoretic deposition, where a solution of the substituents of MIL88B, iron and terephthalic acid, are plated onto a stainless steel cathode using a voltaic cell. So the traditional synthesis route for MIL88B requires a 24-hour holding period, and it uses heat to catalyze that reaction. So the possibility of using a voltaic cell to plate the MIL88B onto a stainless steel cathode in a matter of minutes would be really helpful for the rapid commercial synthesis of the MOF. And here's a table displaying some of the different variables that can be changed in electrophoretic deposition testing. There's a lot of potential experiments that can be run to determine the optimal voltaic apparatus. So for instance, changing the current can have a diverse impact on the size and structure of the crystals plated, along with the duration and concentration of the MOF substituents. In addition, the presence of electrolytes may be needed to ensure that there's enough current passed through the solution in the liquid phase to enable plating to occur. But I'm excited to work on this project as there are currently not a lot of research on the rapid synthesis of MOFs, and I hope to design a faster synthesis route for MIL88B as this will aid our project immensely. And then that's everything that I've been working on since December of last semester, and our lab is really looking forward to continuing this research and hopefully getting a few more papers out there regarding MIL88B. So at the end here, I just wanted to thank STEMnet SoCalGas Fellowship once again for making some of the research described possible and for the opportunity to present it here. So with that, I'll pass it on to the next presenter. Thank you.
Thank you, Ethan, for your presentation. Our next speaker is Erica Esteban from Cal State Fullerton, local and long-range structural investigation of a solid solution exhibiting tunable thermal expansion.
Thank you so much for that introduction. So again, my name is Erica Esteban, and my project involves the local and long-range structural investigation of copper 2- X and X pyrophosphate exhibiting tunable thermal expansion. So my project involves two types of thermal expansion. First, positive thermal expansion is the increase in unit volume as heat is applied. And this is commonly seen when we have a difficult time opening a spaghetti jar, where when we run it in hot water, the metal lid is able to expand more than the glass jar. In contrast, negative thermal expansion is the decrease in unit volume as heat is applied. And we commonly see this where we have an ice cube melting. Now, considering the problems that are associated with positive thermal expansion, we want to study how negative thermal expansion materials can act as thermal compensators for these problems. So with my starting material, which is copper pyrophosphate, it exhibits negative thermal expansion. We want to chemically substitute zinc and retrieve a final product of zinc pyrophosphate, which exhibits positive thermal expansion. Now with this, we want to study the structural changes that are occurring between these two thermal expansion ends. So for my overall plan, first, I have optimized the synthesis of my samples. Then I was able to confirm phase purity and investigate the lattice parameters using X-ray diffraction techniques, and also use these phase pair samples and send them out to collect pair distribution function data. So what exactly is X-ray diffraction? X-ray diffraction is a characterization technique used for solid state compounds, specifically crystalline materials. And with this schematic diagram here on the left, we have an X-ray source shooting a beam focused by a collimator, and this hits the crystalline material to obtain this X-ray diffraction pattern. And with this X-ray diffraction pattern, based on the range that you're using, for us, we're using the 2 theta range, we retrieve a slice of this data and study these peaks using rate flow refinement. So the 2 specific X-ray diffraction techniques that I use in the lab for my project are the lab diffractometer, and this is in-house on campus, as well as get high resolution data sent to the synchrotron. And you might be wondering why I included the whole facility. Well, this whole facility is actually the XRD, allowing the electrons to be accelerated around this entire ring. So for my methods, I use a 3-stage process of solid state synthesis. And first, I want to remove the unwanted materials. So I collect the starting materials, and based on the X factor that I'm working with, I weigh it to its appropriate amount, and I grind it on a mortar and pestle to homogenize the mixture. Then I pack it onto a pellet press for the atoms to diffuse together. And then I apply this to a low temperature heating on the furnace to react out any of the unwanted materials. Then I regrind this solution that was heated again onto a pellet press, mortar and pestle, pack it on a pellet press, and then do a final higher temperature heating to prompt the reaction of the X factor that I want. And then finally, I apply this towards data analysis, where I first apply it on the lab diffractometer to obtain low resolution data, and then send it out or collect data at the synchrotron. So for my data, first I use Rietveld refinements using the lab XRD to get a sense if I have made the right thing or not. And with this, first I look at my data, which is represented in the black circle, and compare it to this model structure, which is colored here in red. And we try to look if my data is able to overlap with the model structure, which it does. But we also investigate if there's any significant differences that might sway my data away from looking like the model structure. However, based on all the X factors that I have made, I can get a sense that I did make the right thing. And then we also want to compare the structural changes. So looking at the volume here, I was able to see that as I added more zinc, there was an overall decrease in volume with a sudden jump of increase in volume as we had 100% zinc. And this is quite unexpected as zinc is larger in size than copper. So I was expecting more of an increasing trend for volume. So with this unexpected result, we wanted to further look at the specific structural changes. So based on the structure here, for the A-axis, we saw that there was a decrease in size. For the B-axis, we saw there was an increase in size. And then for the C-axis, there was a decrease in size. Now, for the beta parameter, which is the angle between A and C, we saw there was a decrease in angle. And then again, for the volume, there was an overall decrease in volume. Now, we also wanted to obtain high resolution data, which we acquired. I was able to acquire in person at the Argonne National Laboratory, and I wanted to confirm phase purity of my sample. And the reason why this is high resolution data is because with the acceleration of these electrons, we're able to get better peak widths to be observed. So again, comparing my data, which is the black circle, to the colored line, we were able to see that overlap between my data and the model structure, showing that I can confirm phase purity for all of the X factors. Again, looking at the structural changes for the high resolution data, we saw that for the overall volume, there was an overall decrease in volume as more zinc was being added. Again, with that sudden increase in volume when we had 100% zinc. And also for the structural changes in the lattice parameters, we saw that, again, there was an overall decrease in size for the A and C axis, but an increase in size for the B axis. And then for the beta parameter, we saw a decrease in angle and again, a decrease in volume. So what is significant is that although we had low resolution data and we were able to compare it to the synchrotron data, which is high resolution, we still saw the same structural changes where there was an overall decrease in these parameters and this unexpected increase in the B-axis parameter. So considering this, we also want to study this using pair distribution function data. And pair distribution function data allows us to obtain the distance between two atoms, and it also gives us more information about the structure of the samples that I have made. In addition, the reason why I want to collect this data aside from XRD data is because these two are actually complementary, where PDF can't allow me to confirm phase purity, but it will also instead give me more information about its local structure rather than just average structure that XRD techniques provide. So what exactly is average structure versus local structure? If we compare the crystalline material, which is what I am working with, a repeating unit cell, and compare that one unit cell to a person in a family, for average structure, we would only be able to obtain the average age of the person in the family, and we won't be able to know what the age of each family member is, meaning the structure of the overall crystalline material will only be able to get the average structure. However, if we look into local structure, which PDF provides, with our family, we'll be able to see what are the individual ages of these family members, as well as compare the difference in age between the youngest family member to the eldest family member, or any other ages in between. Again, with my crystalline material, we'll be able to see what are these specific disorders that we weren't able to see with the XRD data. Now, we were able to acquire proposals for 2 of these sources, also considering that collecting PDF data is quite competitive and limited, as there are only 3 facilities that we can collect this data in the country. However, we haven't been able to obtain that data yet, and we look forward to retrieving that data. And in conclusion, I have been able to synthesize my samples from X factor 0 to 2, as well as test different copper sources. I was also able to confirm phase purity of my samples, and we're ready to submit my samples to be observed under neutrons. So for my future plans, we have planned to obtain pair distribution function data, again, to observe the local structure. We also plan to study these with neutrons, because neutrons are not systematic, as copper and zinc are only 1 electron apart. So studying them with neutrons would be beneficial. And also we want to obtain neutron diffraction data to get the true copper and zinc ratio. So I'd like to thank the following acknowledgements for their contribution to my project, as well as Dr. Cooley for her mentorship. And thank you. I look forward to your questions.
Great, thank you, Erica, and certainly everybody who spoke today on their wonderful research. We do have about a little time for a Q&A session now. So even speakers can ask each other questions too. So we'll just open it up. You can just, if you have questions, by all means, just raise your hand either physically or in from a pictorial perspective. So we're open now to questions. Don't be shy.
I do want to say, everyone who presented today, you did great. Your research was fascinating, so good job. You deserve it. So.
Yes, Dr. Cooley.
Great job, presenters. So I guess this question is for any of the presenters that would like to comment on this. If you can say briefly, I think one of the presenters did say kind of what she learned from this experience. There was kind of a negative result that you got, and I think it's really fascinating to hear how researchers think about, okay, what I expected to happen didn't happen. What do I do now? Did any of you have those moments throughout your research?
I can begin by answering that. Absolutely. I think, that's part of both the frustration and excitement of research. Most recently, it was with a 3D printer. Oh my goodness, my camera died. How awful is that? Let me turn it back on. I experienced it recently. It was the 3D printers we were using for our my camera keeps turning off. I'm so sorry. I could plug it in after this response. Basically, we found that the software we were using, it was just too difficult to access. And so we decided to make the change to another 3D printer, and we got it running quicker than we would have if we stuck with the other one. So what was originally a failure kind of ended up being a better solution. And so absolutely, I open that solution space. It's pretty, it's frustrating, but I think you can learn a lot from those. Thank you for your
Yeah,
question.
I could also I understand what that's like as well for our project too. We were supposed to go out to the field to collect data for our set, but it was during the time of the floods and stuff, and the road to get to our site was like, you know, it was flooded. There was no way for us to get through that, so we had to compromise, and we went through different data sets of unpublished work and published work to try to complete to try to get the data we needed for the project. So, you know, that was something we had to work through, but it all worked out in the end, so that was nice.
I can add on to that too. For my research in specific, a lot of it revolves around growing crystals, right? And we're talking about the nanoscale, so a lot of it can only be seen under an electron microscope, right? But I think it's really cool because sometimes, you know, you can't physically see what's going on until you've already grown them, and then afterwards you'll be looking at your sample, you know, under microscope, and you'll find something, you know, you probably that wasn't your intended target, but sometimes it'll be more interesting than what you were actually trying to get. You know, you can really learn a lot from your failures. And I think that that's really, you know, what keeps you going in research is trying to learn like everything other than what you're trying to get, you know, so.
Inputs from anyone else about their experiences?
I think also to add on, aside from failures, we might also come across frustrations where we can't have answers right away. However, it's also good that we should be motivated and also open to reading other literature who will give us insight on finding ways we can solve the problems that we come across.
A previous faculty member, when I was many years ago, an undergrad, said a couple hours of time in the library, and I know your library is now on your phone. You don't actually physically have to go to the library and make Xeroxes. You probably don't know what that word means now. Okay. So a couple hours in the library will save you weeks at the bench if you were an experimentalist per se. So that's something that is probably still true, but just substitute the word library for something else. Okay. Okay, so for Trent, maybe you can read that. Trent, there's a question in the chat.
Oh, yes, absolutely. Thank you. So one of the reasons why we chose the Arduino is that we can easily access it through a COM port in MATLAB. We generally don't have issue with the amount of data that we can pull through. Usually the baud rate is high enough. If it does come to the point where we need a little bit more bandwidth in the data that we're collecting, we can switch to a Raspberry Pi and experience larger bandwidth. But for us, it's an Arduino Mega that's running the controllers in the printer. And it's nice because all the firmware is open source. It utilizes the Arduino IDE, and these are things I'm pretty familiar with. So it allows that control and communication to be pretty effortless. And it's one of the reasons why we got it online so quickly over the other printer that was using proprietary software and firmware to promote that communication. Thank you. hopefully that answered your question too.
So I have a question. Was this for all of you who spoke today? Is this your first research experience, and how long have you been doing research in the lab that you're currently in?
So for me, this isn't, I've been doing this research for the last few semesters, but I do want to say that I'm honored to have this experience and this opportunity because it allows me to focus on my research. And I think sometimes to have this financial support to really focus on things that we like feels somewhat gluttonous, but it's not. And so thank you. I'm really honored to be able to have had this experience and share it with all of you.
I started working in Dr. Pratt's lab last summer. We did it was like a month-long summer undergraduate program. And this is how I got introduced to this research, and I've been doing that since then. So a little bit close to a year now. Not quite there yet, but getting close to there.
Yes, for me personally, I joined my lab around last winter break, but I actually got to start doing some experiments very recently, so this semester. And though I initially thought that it was going to be extremely stressful to take on like such a project as my first semester, this opportunity was really just an excellent opportunity to start really getting into the main research components of what I was doing. So it basically functioned as an introduction with the prompt that I was given, and I got to actually start to write the first scholarly components of like my report and everything. So I would say that even though it seemed a little bit daunting at first, it was 100% worth it.
For me, this is my 4th semester working on my project, but sadly it also is my last semester as I am graduating. But I do look forward to the person who will be taking over my project and seeing how much my project does grow.
So I've been doing research for a little over a year now. I started when I was a freshman, so I went into it having no idea what anything was at all. But I do appreciate the opportunity to be able to present it here. I really like being able to share what I've been working on with other people. I think that's really cool, and I do appreciate the funding and opportunity that this the program allows, because it definitely does motivate you to focus and actually get some results from your research. So thank you guys.
This was my first time doing research, and I've been doing it for this semester. So I'm really grateful for this opportunity so I can learn more about how to actually do research for the first time.
Well, I'm glad you all enjoyed your time and will continue. Erica, I know you're going to be moving on to brighter waters, but I'm glad you enjoyed, all of you, your time doing research. And it's certainly in the beginning, it could be trying because you don't know what you're getting into. New experiences can be that way, but the days go and move on, and eventually you become that one of those senior in your group that the younger students turn to, and then it's time for you to leave. But you wear different hats over your year or a couple of years as a research student. You're first a beginner. You're learning. You're maybe working on a project with some more advanced students, and then you transition to at one point being that person who has their own project, who doesn't need as much direction, and then you become quite senior, where you may serve as mentors. Do any of you do that, where you have younger students that work under your auspices?
We definitely, because the research is so multidisciplinary, we do have younger students that are part of Dr. Flores' team that we bring on every semester. And I think that is part of the challenge, but it's fun. As scientists, you know, we're here to transmit knowledge. We're here to safeguard the things that we know and ensure that other students and other scientists have access to it as well. And so, yeah, absolutely. You know, we're teachers in a sense, and I think that's pretty neat.
Well, I appreciate your time and your effort and all of your hard work. And, you know, I still have a small lab, even though I'm at the chancellor's office. I have 6 students working, all undergraduates at Cal State LA. I've probably had about 150 to 170 students over my many years. And it's always great to see how they come in very green, and then when they're ready to graduate, they're so good that I'd like to keep them, but they need to go on to brighter waters, to grad school or med school or dent school or vet school or wherever they go. And so now I kind of ping them, now that some of them are doctors or several of them are professors, and ask them, you know, about their experiences, and now they're mentoring students. So now they're in my place. So it's always great to see, you know, those people that came in at 18, 19 years old, who are now 35, 40 years old and have students working with them. So, you know, it was great seeing you all today. You did a great job. Best of luck to all of you as you progress in your studies, both within the CSU system and outside. To everyone else who's been in attendance, thank you for supporting CSU's best and brightest. And I certainly appreciate Southern California Gas for funding. We've had this funding for a few years now, and hopefully it can continue on and grow, because certainly research is a very high impact practice for undergraduate students. And it's better to stay within the school grounds and have that educational taste than working outside a campus. You don't have to drive, and you could study and have a community of other students, not only in terms of research, but also in your classes, which helps too, as I'm sure you've all learned. So once again, I thank Monica for helping in coordinating all of this, and certainly SoCalGas, and to all of you. So to all of you, have a great weekend, and have a great day. Take care.
Thank you so much.
Thank you.